Hello everyone, my name is Guilherme Neumann and I'm a PhD student at Humboldt University of Berlin in Germany. And today I'm going to present a part of my PhD thesis which is entitled Content and Design of the DSN200K SNP-chip, a new tool to assist genetic diversity management in an endangered German cattle population. This is a project done in partnership with the University of Gießen, also in Germany. And the main idea today is that I present some insights and technical information on how we designed our customized SNP-chip. So I'm very glad to be here. I'd like to thank Tamo Fischer for this invitation, and also thank you for the interest on hearing from me. So very brief description on DSN. So DSN is just a shortcut for the actual name in German, Deutsches Schwarzbuntes Niedersrundrind. So in a free translation to English, it's German black pied. It's a Bos Taurus dual porpoise breed, which means we use it for both milk and beef. And it's a black and white coat, genetically horned, as you can see. And it's considered the ancestral population of Holstein cattle. So those are animals from the North Sea region, which were exported mainly to the US, and where they were used for very specific breeding programs, generating then Holstein. So nowadays we have DSN producing from 7,000 to 8,000 kilograms of milk per lactation, which is about 2,000 kilograms less than Holstein. And because of this, DSN has been largely replaced by the Holstein cows in the last years, generating a current population size of about 2,500 animals. And for that reason, because of this small population size, this is classified as an endangered breed according to the Society for the Conservation of Old and Endangered Livestock Breeds. So it is also an official gene reserve subsidized by the German government. So the idea is to keep DSN as a gene reserve, as a pool of genes that could be used for genetic rescue in Holstein in the future if needed, for example. And it's also a way to conserve genetic diversity in Bos Taurus as a whole. So, so far what we have been able to do is that we detected some variants and candidate genes in some important traits for DSN. Those are milk, mastitis resistance, and endoparasite infectious resistance. Those are mainly traits which are important in a context of having DSN inside organic farm. And also we were able to develop an economic lifetime net merit for DSN, which makes us able to evaluate the value of DSN in economic context. We also were able to impute to test strategies for imputation in small populations such as DSN, which is quite tricky when you don't have a huge reference panel. The only source of genetic information that we have been using into the moment is the Illumina SNP-50. So what we normally call the Illumina 50K, and how I'm going to call it here today. And from the 54K variants that we have, normally after a call rate and manual allele frequency filtering, we only have about 36.9K in DSN for more than 2,000 animals. So for that reason, we lost some information. And we also miss important information regarding functional variants, and we have a shortage of DSN typical variants. And of course, an absence of rare variants, so variants which are in a lower frequency. For that reason, we realized that we had a gap when thinking about the application of conserving DSN and as having a gene reserve. So it might be enough for certain applications, but when we talk about the gene reserve, we are actually talking about genetic diversity management and ultimately at maintaining heterozygosity. So our main goal is to maintain heterozygosity, which ameliorates inbreeding depression and loss of genetic variation at loci that may become of importance in the future. So it may become, right? So this is the idea of conservation. It's a unknown future, ain't no trades, and that's the beauty of it. And it can actually conserve Bos Taurus throughout those local breeds. But how really being aware of those loci that we don't know yet. And also we could be able to avoid genetic drift, which prevents deleterious recessives, so for example, rare diseases alleles, from drifting to a higher frequency, and also prevents random drift of important functional traits. So with that in mind, we decided to customize a SNP chip for DSN. So considering both whole genome sequencing variants and Illumina variants, so it means all those 36K variants that we had, we decided to keep using those, but adding variants that might be interesting. And those are DSN typical variants, functional variants, diverse findings, and differences between DSN and Holstein. With that, then you would be able to approach more effective genome-wide association studies and work with marker system selection, even though it's tricky because of the population size, and also work with genetic diversity management, our main goal, with mating suggestion, parentage control, and so on. So what we needed first was an initial data set of variants that we would have in DSN over the genome. So we decided to sequence 304 DSN animals. Those were systematically selected. All insemination bulls that we had, which are 47, and 257 cows. Those were sequenced in the Illumina NovaSeq. And we process all this data according to the guidelines given by 1000 Bulls Genome Project. And also the filtering was done according to 1000 Bulls, where you work with the variant recalibration, which is a machine learning approach done on the GATK software. So afterwards, we had a list of 20 million high-quality variants. Those were 18 million SNPs and 2 million indels. And that's interesting to remember here that from this time on, we are working both with SNPs and indels. Afterwards, we also defined haplotype blocks. So of course, we couldn't work with these 20 million variants. So it would be nice to have a genetic distance understanding of those variants. So for that reason, we defined haplotype blocks in Blink, and those were 186K. So at this point, we had to decide, of course, which platform we were going to use. That's why I am here, because we decided to use the Axiomine design from Thermo Fisher. This is a very personal decision, of course, and especially the size of the SNP chip. It depends on how many animals you have available for genotyping and how many markers you want. So the more markers you have, the cheaper it gets. So yeah, I'm not going to discuss it here. What I can say is that the one that we selected has about 96 samples per chip, and it's up to 200K probe sets. So what you need to know is that what you give to Thermo Fisher is a forward strand. So you give a sequence of 7 to 1 bases where you have in the middle your variant, so the variation. And yeah, Thermo Fisher will tell you how likely it is that this probe is gonna work at the end. And even not only for the forward strand, but also for the reverse strand. And what you need to know as well is that sometimes when you have GCs and ATs variants, then you might need 2 probe sets per marker, which is a technical aspect of Thermo Fisher technology. So at this point, you might wonder, okay, but probably how long it takes. It would take years or whatever. And actually, this is how long it took to our case. For us, what took the most was the whole genome sequencing. This is, of course, the most time-consuming part. For us, it was about 4 months. And yeah, we had 300 animals, but it depends if you have your data or not. And the array design took about 3 months, but it could actually have taken less. It depends on how clear is your criteria and how aware you are of what you want. So just to give you a brief understanding of how you interact with Thermo Fisher. So you have a first meeting with them where you discuss the market selection strategies. So they tell you what you should keep in mind and what is just advisable to do. And you agree on a list of variants that they are gonna proceed with in silico scoring. So this in silico scoring is a machine learning algorithm that they have on their own, and it's based on their database and also on other aspects, and they tell you how likely it is that this variant is going to work at the end. And then they also advise you on very specific topics that is important to your work. And in our case, they helped us with the decision about the parentage panels and also with mitochondria, because we wanted to have as many variants from mitochondria as possible. And so they gave us all variants that they have in their database for mitochondria. And also they advise you at the end, as this last step, how many probe sets you should have per marker. As I said, ATs and GCs, you normally have 2, but in some cases for important variants of yours, you may say, no, I want to have also 2 or even more, and they help you with this decision. And as soon as your SNP-chip is done, the design is finished, so of course you go for genotyping, and then they also help you with the development of the library files. Actually, they do this for you, and you might ask them for some changes if you want. And then you use these library files in the Axion analysis suite, which is the software for the genotyping. So first things first, what we need to start doing is, of course, to reduce the number of variants that we have. So we had 20 million variants. So the first thing that you need to do is to remove all variants that are clustering with another variant in a neighborhood of, let's say, 30 base pairs. It means all variants which had another variant which is within 30 base pairs should be removed. So this is what Thermo Fisher says. And in our case, since we have too many variants anyway, so we decided to remove everything within 35 base pairs, which means we would have at the end probe sets of 71 base pairs with only our target gene, our target variant, nothing more. However, in the case we had like important variants of ours, that like Jiva's results that we still wanted in the chip, so we allowed a clustering within 20 base pairs. And yeah, Thermo Fisher says that for diploid organisms, it's still possible within 20 base pairs. So you can see this is the most crucial step regarding filtering. You lose most of your variants here. So in our case, we reduced the size for 8.9 million variants. And afterwards, we also had all the filtering thresholds, like the coverage. We removed everything with a lower a coverage lower than 3,000 reads, because we had 300 animals, so we would expect 10 reads per animal, and also the allele frequencies. You may do it with minor allele frequency. Here we used alternative allele frequency, and we removed everything which was less than 5%. However, when we had variants which were DSN unique, so variants that were only occurring in DSN and were not occurring in other breeds closely related to DSN from 1000 bulls, so breeds that we had information, then those variants we kept, variants which were higher than 1% in allele frequency. So this was a risk that we took to have this lower frequency variants, but those are variants unique to DSN and also rare variants, so we wanted to have those if possible. And here it's important to say that we are only talking about variants filtering, so we are not selecting anything yet. So this list of 5.6 million variants was sent to Thermo Fisher, where they actually then do the in silico scoring, so based on forward and reverse trained, and tell you which is the best probe set to use per marker, and they give you a p-convert score, which is this a measurement of how likely the probe set will work at the end. And what they tell you is normally that everything greater than 0.6 in p-convert score is recommended. So we only work at here from now on with recommended variants. Those were 3.1 million variants. And then we finally went to the selection of variants. First thing, we took everything which was most important to us. So all variants from the Illumina 50K, those were the 36K variants, all Jiva's results, all variants which were high, moderate, or low impact predicted by VEP, variant effective predictor, all variants which were DSN unique and had a higher difference between DSN and Holstein, and also all variants from the Y chromosome, mitochondria, and the parentage panels. And we removed all variants from this group that were incomplete LD, because then, of course, we want to reduce the number of variants selected. And afterwards, what we did, we selected all variants per haplotype block that were not covered yet in this first selection. And those were haplotype blocks longer than 1 kb when they were inside a gene or close to a gene of 100 kb. And also all haplotype blocks longer than 5 kb, independently of having a gene or not. And for each haplotype, we took one marker, which were the one with the highest p-convert score and had a minor allele frequency greater than 5%. And if we had variants with empirical data, which means that already we had a proof that it worked before in other SNP chips from Thermo Fisher, then those markers were prioritized. Then we ended up with 182K. And we finally selected manually 16 variants that were filling gaps of 200 kb. So you can see here we have three main points. First is biological relevance, then the genetic distance by haplotype blocks, and then the physical distance. And at the end, we have a SNP chip with 182K tiled, which were from 103,000 haplotype blocks. So here you can see the number of markers per category of selection, those of biological relevance. So you can see 34K from the Illumina, 50K, and 1,000 from Jiva's results, 50K from high, moderate, and low impact. So we have basically a 50K SNP chip only with functional variants. And we have 38K from DSN unique, 55, which a high difference between DSN and Holstein, 321 from Y chromosome, 272 from mitochondria, and 554 all markers from the ICAR and ISAC parentage panels, and then 58 from the haplotype blocks, and 16 from gap filling. And those sum up, if you remove the overlaps between those variants, to this 182,154 variants. So of course, here you have some overlaps between, for example, high, moderate, and low impact variants to the Illumina 50K or to Jiva's, right? But those are the numbers which are more important to us. And you may ask, what about the 200K? Because this is a 200K SNP chip. And actually, it sums up to 203K probe sets, because then we have the GCs and AT variants. And also in some categories, for example, those variants from Jiva's, we also included them twice in the chip. So here you can have an idea of the physical distance, so the distance between tiled variants in the chip. And you can see we only had a long gap. The longest gap was in chromosome 10, which was about 1 MB, and another one which was longer than 200 kb in the chromosome 12. Those were regions which really did not have any variants detected in DSN. And we also have some gaps, or a lot of gaps, in the Y chromosome, but this was because of a problem in the sequencing, where we believe it is a pseudo-autosomal region in the Y chromosome, where we had a problem in the mapping. But the Y chromosome was just something that we wanted to have, but was not priority, because both the Y chromosome and the mitochondria, they are not included in the reference genome of the in the cattle genome, and then that's the problem that you might find. So here you can see how we were able to increase the number of functional variants in our SNP chip in comparison to Illumina 50K. That was one of our main goals. So from in blue, those are the lowest impact variants, and in red, the highest impact variants. So you can see we were able to increase, for example, missense variants to 9% in DSN, and in comparison to bovine 50K, where those are mostly intragenic and intronic variants. Of course, their selection probably had in mind to keep neutral variants, but we wanted to have those functional variants as well. So at the end, just very briefly, how we have been used the SNP-chip. So far, we genotyped about 1,594 DSN animals, and those were 167K variants which successfully culled. So this is about 91%, whereof 86% is segregating DSN at a minor allele frequency greater than 1%. So this culling here, it's not only the culling itself, technically speaking, but it considers also the genotype clustering done by the software, and also all the filtering that you do. We used all the default thresholds by Thermo Fisher, and we also checked the quality per marker. So yeah, we may ask, okay, is this 9-to-1 good or not? It depends on what you wanted. So in our case, we took the risk to have variants that were not culling or even not segregating. And when we look at this table here, we can see the failing variants, they are mainly from the three groups, the high, moderate, and low impact, DSN unique and haplotype block. And we believe the main reason is actually the allele frequencies, which makes sense. If we look at the minor allele frequency from the sequencing data, we see it's quite low. So in DSN unique, it is really low, so it's about 0.03. And in the other two groups, it is not so low, but if you look at the standard deviation, you can see that some variants, they are actually very low in frequency. And also when we look at the p-convert score, those are always greater than 0.6, but they are more or less close to 0.6. And when we look at this distribution here of the culled variants, we can see that the failing, they are between 0.6 and 0.8. Yeah, however, also the ones which are working are also between 0.6 and 0.8 mainly. So this is not the only reason. Probably there are a lot of other reasons behind, and we believe it's mainly because of the allele frequency, like I said, and also the sequencing quality. So we were not so conservative because we wanted to keep as many very important variants as possible. And this is depend on you if you are open to lose some variants, otherwise you should be more conservative about this. But if you want to know a little bit more about the performance of the SNP-chip, this is not the main idea here. You can check out my presentation in session 46, and this is the abstract number. So I believe it's going to be recorded somewhere in the EAP website. So to conclude, we are very glad that we were able to develop a customized SNP-chip for DSN, included very important functional variants. This is a very flexible and diverse selection of variants, and it was not very time consuming. It was a very straightforward solution. Of course, there might be other solutions for genetic diversity management, but we believe that this is one of the most cost-effective solutions. And the most interesting thing about it is that we are able now to update the failing variants. So we could, for example, take 10K variants from the SNP-chip which are not so nicely working and just replace those with variants with a stricter minor allele frequency and quality control thresholds. So that's the nice thing about it. And I hope you enjoyed and you learned something that it might help you with your design. Of course, I'm able for questions. And I would like to thank the DSN breeders and RBB for supporting us with data and material from DSN animals, and also Thermo Fisher for the invitation today, and also from the support given during all the design of the SNP-chip. And also thank of course, the funding from the Federal Ministry of Food and Agriculture of the German government. Thank you very much.